Hello students, welcome to EPZ Pathshala. I am Dr. Nirupama Trahenpati from Institute of Liver and Biliary Sciences. Today we are going to discuss the molecular cellular therapy from paper Molecular Therapeutics. Purpose of the cell therapy is to introduce cells into the body that will grow and replace damaged tissues. Cell therapy differs from conventional stem cell therapy in that the cells injected into the body. In cell therapy, we already have the differentiated cells like muscle cells, gland cells, neuronal cells, whereas in conventional stem cell therapy, cells are utilized which are undifferentiated, usually embryonic cells. Cellular therapy definition is that cell therapy called cellular therapy. Why? Because cellular material is injected into a patient or in a human body. For example, T cells which are capable of fighting cancer cells via cell-mediated immunity may be injected in the cellular therapy. Cellular therapy is transplantation of human animal cells to replace or repair damaged tissue. This is a technology where we rely on replacing diseased or dysfunctional cells with healthy and the functional cells. Cell therapy may be applicable to some types of diseases like cancer, neurological diseases such as Parkinson, amyotrophic lateral sclerosis, spinal cord injuries, and diabetes. A great range of cells can serve in cell therapy, including blood and bone marrow cells, mature and immature solid tissue cells, pluripotent stem cells, and T cells and dendritic cells. Like we can use the pluripotent stem cells. One of the advantages of the pluripotent stem cells is the availability to create cell models for diseases. Pluripotent cells can be in vitro generated through the fertilized egg. It can deliver it to the embryo, and in embryo with the blastocele, and we can isolate the pluripotent stem cells. It can be differentiated into the blood cells, cardiac muscles, and neural cells. What are the cancer cell escape mechanisms? Cancers or the tumor generally escape with definitive mechanisms. They have the defective antigen presentation, or either tumor-induced inhibitory checkpoints pathways are against affected T cell activity are damaged, or there are regulatory T cells present and the cancer tumor mass is too much, or the myeloid-derived suppressor cells are too much, and through all these kind of cells and mechanisms, cancer cell therapy, it is important that we utilize the functional cellular therapy to replace these damaged cells. Secretion of immunosuppressive cytokine is one of the mechanism where the cancer cell escape, such as transforming growth factor beta or interleukin 6 or VEGF is more present in the cancer cell mass, and that is the mechanism where the cancer cell is making the escape mechanism through these cytokines. What could be the strategies for the cellular therapy? There are the adoptive transfer of immune effector cells. These therapies are called immunomodulatory cell therapies. These effector cells can be from the innate immunity like NK cells adoptive transfer, macrophages adoptive transfer. Most popular is dendritic cells, effector cells from the innate immunity adoptive transfer. In the adaptive immunity, basically the T cells and the B cells are popular therapies which are being used in the diseases and in clinical trials. There is the adoptive transfer of cellular therapy, which is called adoptive transfer of immunity. These adoptive transfer of cell therapy from the bone marrow or the total isolation of peripheral blood mononuclear cells. Right now, I will give you one example. We have to give the bone marrow transplant or the peripheral blood mononuclear cells from donor to hepatitis B patient, which is chronically infected with the hepatitis B. Now we will isolate bone marrow or the PBMCs from the donor, which is HLA identical, HLA matched to the patient. We will isolate the total PBMCs from the whole blood and infuse into chronic hepatitis B patient. This is the total cell therapy and called as adoptive transfer of immunity. There are many forms of cell therapy, like as I previously told you, whole blood transfusions, platelet transfusions. We can isolate the packed red cells and can transfuse. Bone marrow transplant is the one of the option, and in fact even the organ transplants also comes under forms of cell therapy. There are many tailor-made cell-specific therapies, like in the case of cancer. In the case of cancer, we know that tumor mass, the whole cancer mass, have different population of the cells. Cells from the tumor, they are called as the cancer cell. Cells which are present along with the T cells, T lymphocytes in that tumor mass, is called as the CTLs, cytotoxic tumor T cells. And the cells with the cancer cells, they have the cancer specific cells, which we called cancer stem cells also. So many kind of cells are present in them. Now we have to generate the cellular therapy against particular cancer stem cells or cancer specific cells or cancer tumor infiltrated cells. So these therapies will be the specific, and tailor-made therapies can be generated, and this will ensure that recurrence of tumor mass or recurrence of cancer should be prohibited by the cell-specific therapies. Adoptive immune therapy or adoptive cell therapy, which basically recruit adoptive T cell transfer, or it recruit the NK-mediated cell therapy, which kills the NK-mediated killing of the cancerous cells. It can be cellular therapy with the combination of cytokines like IL-2 and interferon alpha combination with the chemotherapy. So three kinds of cellular therapies are most popular in the diseases, which explains how the T-cell immunotherapy for the cancer, especially the liver cancer, which is called as the hepatocellular carcinoma, is important. As you know that liver cancer or the hepatocellular carcinoma ranks the 6th most common cancer in the world. HCC can be prevented and treated, and it is a great concern how to treat specifically only the HCC, not the normal cell of the body. HCC generates the specific immune responses, and especially in cancer, there are tumor-associated antigens which are presented on the tumor mass. And seeing the tumor-associated antigen, there is an activation of T cells via the T cell receptor against these tumor-associated antigens. Above all, the activation of T cells, there are the suppression methods which continuously grow in the cancer and make the cancer mass to grow randomly. These are the suppressive factors, and they suppress the activated T cells. These are the TGF-beta mediated suppression or the regulatory T cell mediated suppression. Therefore, treatment of hepatocellular carcinoma is important. Other than the chemical or chemotherapy, immune or cellular therapy become the second branch for the HCC treatment. A scene of immune tolerance and inactive tumor-specific T cells caused by the immune evasion mechanism lead to the loss of capabilities that immune system cannot recognize TAs and effective immune response against tumors. Therefore, the cellular therapies are being generated for the HCC. There are genetically modified T cells can be generated for the as cellular therapy. Like in hepatocellular carcinoma, like many other solid tumors, it is composed of heterogeneous population of cells, which also contains the cancer stem cells. Although the process of hepatocarcinogenesis is multi-causal and multifactorial, HPV and HCV infection are the preliminary infections which cause the liver diseases or the hepatocellular carcinoma. Other than these hepatic viral infection, metabolic liver diseases are also leading to the HCC. Now, how we can rectify these HCC through cellular therapies? Genetically modified T cells are being cultured and made through T cell clone from patient who is already infected with HPV. Now this cloning of TCR DNA or T cell receptor DNA, which is packaged into the another virus, and now this virus, which has the genetically modified T cell receptor, transduced into the normal T cells, and these transduced T cell receptor along with this T cell can be infused into the patient for the genetically modified T cells, which will be the specific to the hepatitis B virus T cell receptor specific, and it can work much better way than the activated T cell which is present in the tumor mass or the cancer of the liver. Other than the therapies, why the cellular therapies are important in HCC? It can have the remarkable tumor clearance. T cell cellular therapy for HCCs are important as the autologous T cells you can isolate from the patient. Those T cells can be manipulated via the genetically modified TCR, or the total cells can be manipulated, and those T cells can generate now better IL-2 secretions or the cytokine secretion and can infuse back to the patient for HCC clearance. As an emerging method of tumor clearance, this is the most effective and selective target cellular therapy for tumor cell evasion. Various tumor immunotherapy techniques associated with the T cells have been developed during the last 2 decades and brought excellent outcomes. There are different kind of activated killer cells like lymphokine activated killer cells. From where lymphokine activated killer cells comes? We can isolate the lymphokine activated killer cells through the peripheral blood leukocytes, then in the patient with the cancer, and then we can activate in vitro by IL-2, leading to the enhancement of effector lymphokines, and these cells are cytotoxic to autologous fresh solid tumor cell in manner unrestricted by MSC, which was earlier many years ago, like in 1980s was reported. So these lymphocytes are called lymphokine activated killer cells. It has been reported that lymphokine-activated killer cells therapy could significantly prolong the survival time of liver patient or liver failure patient or liver cancer patient, but failed to decrease this liver cancer mass. However, LAC cell therapy for HCC has induced objective responses in minority of the patients, but it produces considerable side effects also, like dose of recombinant IL-2 become more and it become toxic. In view of the limited efficacy and increasing doubts whether lymphokine activated cells indeed cause the observed responses, the initial enthusiasm for LAC cell has been transferred to other immunotherapies. Other than the T cells, there are the tumor-infiltrating lymphocytes, which includes T cells, B cells, dendritic cells. Total pool of total infiltrating lymphocytes have the impressive role to clear the tumor. Tumor-infiltrating lymphocytes, which is called as TILs, which play a positive role in survival of HCC patients. They can recognize the tumor-associated antigens on tumor cells and then trigger anti-tumor responses, therefore isolating, stimulating and expanding the total TILs in vitro from HCC patient and then infusing into host could theoretically kill the tumor cells. However, many discoveries happened, included the recent discovery of the Stephen M. Brunner, who indicated that tumor-infiltrating TILs produce IL-33, effector memory CD8 T cells, and they are independently related to prolonged HCC patient survivals. Clinical trials utilizing autologous TILs in patients with HCC following tumor resection have indicated that reinfusion of autologous TILs after activation and expansion in vitro can be successfully performed with low toxicity. As this figure shows you, tumor-infiltrating lymphocytes as therapy, as cellular therapy, highly personalized treatment. It is expensive, labor-intensive, and requires high laboratory expertise. To crop the benefit of tumor-infiltrating lymphocytes, in the clinical trials in early stage of cancer, it is acting as adjuvant therapy. What happens along with the chemotherapy or the radiotherapy of the patient, from the tumor mass, TILs are isolated. Now these TILs in vitro activated and expanded. Now these TILs are being transferred into the patient, which they release the cytokines, and that cytokines help them to reduce the tumor mass, and therefore along with chemotherapy and radiotherapy, this is called as the adjuvant therapy or adjuvant cellular therapy. There are other kinds of cells like cytokine induced killer cells. These are called 6 cells. They are mixture of non-MHC restricted T lymphocytes comprising CD3 positive 56 positive cells or CD3 negative 56 positive NK cells. These are the main effector NK cells and which are rare in cultured peripheral blood. Now, capitalizing on patient instinct to eradicate tumor cells via stimulation with cytokine, these cells should be cultured with the IL-2 and OKT3. These 6 cells could be converted from peripheral blood mononuclear cells to the LAC cells or the 6 cells. Now, these advantageous aspect of these 6 cells is a favorable option in cancer cellular therapy. Other than lymphocytes or the TILs, there are cytotoxic T lymphocytes, which also recognize the tumor-associated antigens by APCs. When TAAs or tumor-associated antigens are processed and presented to the MHC by antigen-presenting cells, T cells are activated. This activation is triggered by two signals, one of which is from the binding of TCR to the MHC bound antigen. Then signals are magnified or counteracted by co-stimulatory molecules. Now these cytotoxic T lymphocytes, which could be the CD8 positive alpha beta T cells, could recognize MHC molecule present on TAs and release granzyme B or the perforin to lyse the total tumor cells. However, the downregulation of MHC, lack of co-stimulatory molecules, or existence of suppressor immune cells in the tumor mass like Tregs or myeloid-derived suppressor cells, which make difficult task for the CTLs to reduce the tumor mass. Therefore, the autologous cytotoxic T lymphocytes can be isolated, expanded, activated, and reinfused as potent cytotoxic lymphocytes in the patient for better reduction of tumor mass and called as adoptive therapy along with the chemotherapy and the radiotherapy. So, as I told you earlier, that T cell receptor can be engineered. We can isolate the T cells of the patient. That T cell receptor can be changed, which is very specific to the HBV, hepatitis virus, or to the CMV, or the any kind of infection, and then reinfuse into the patient. So these chimeric antigen receptors, which is called as the CARs, along with the T cell with the ability of recognize defined or specific antigens, become the important cellular therapy. CAR T cells can distinguish an overexpressed tumor cell surface antigen directly. Now the another benefit of CARs select tumor-specific antigen which is not significantly expressed in the normal tissues. So they only going to lyse the tumor mass which is associated with the tumor-specific antigen or tumor-associated antigen, and they are not going to lyse the normal tissue cells. So CAR-specific T cells is also the one of the good cellular therapy. However, all these cellular therapies are under the supervision in the clinical trials. As explained in the previous slide, CAR T cells for the liver diseases. We can isolate the T cells from the patient blood through leukapheresis. Then there are the CAR which can be generated as the second generation anti-CEA scurvy CD28, which is the co-stimulatory molecule, and CD3, which is the receptor of the T cells. Both are cloned together into the retrovirus. Now the second stage that when the CAR T cells should be expanded. Now these CAR T cells are activated and transduced with the modified CAR T cells. Now the normal T cells, when they are transduced with modified CAR T cells, they are now being expanded in vitro. CAR T cell adoptive transfer is the third stage of this cellular therapy. Through the arterial access, there is a procedure where the infusions of CAR T cells will be delivered into the hepatic artery. Further, the patient will be monitored for disease response, and bone marrow biopsy will be done to see whether the T cell suppression has gone away or not. It can be checked with the interferon gamma levels, and we can track engineered cells to kill the tumor mass or the tumor cell. However, there are the drawbacks of these chimeric antigen receptor engineered T cell or the cellular therapies. What is the drawback? That tumor associated expansion in normal tissue varies. Therefore, CAR modified immune cells attack the tumor tissue will also cause damage to normal tissues or organs. It's not very safe cellular therapies, and there are problems of insertional mutagenesis in these CAR T cells, which can further make the tumor cell surface antigen directly or indirectly and can destroy the normal cells also. Other than the T cells, there are the cellular therapies which target the natural killer cells. Like natural killer cells are the second player in the cancer biology, which they can utilize to reduce the tumor mass. Now there are different kind of natural killer cell cellular therapies. Autologous or allogenic natural killer cells are usually obtained from the patient or the donor blood. They can also be derived from the bone marrow or umbilical cord blood or human even embryonic stem cells. Now these therapeutic NK cells can be matured through some cytokines, or NK cell progenitors can be matured into the mature NK cells and can be inoculated together with other cells as part of the cellular therapy. And however, this cellular therapy should be pre-enriched, like the mature NK cell should be in good population in number, and those are the pre-enriched process. Now enrichment of NK cell is generally achieved by magnetic depletion of T cells or selection of specific CD56 positive cells. Then now these enriched NK cell can be expanded in vitro in culture for 1 to 3 weeks in the presence of IL-2 cytokine or without feeder cells which experienced or engineered to express cytokines and co-stimulatory molecules. Depending on the different protocols, finally NK cells should be matured, expanded, activated, and show the variable purity, that whether it is a 99% pure, 80% pure or 90%. These all factors might include a substantial proportion of even contaminating T cells. So those T or B cells should be again depleted, and residual enriched mature NK cells should be infused to the patient for the better reduction of cancer or the tumor mass. CAR-based natural killer cell therapy. CARs can be engineered in autologous or allogenic NK cells or in NK cell lines such as NK92. CARs are designed by the fusion and antigen binding domain with the hinge region, a transmembrane domain, and one or more stimulatory molecule. Each CAR has the CD3 zeta chain as its main signaling domain. Additionally, 1 or 2 co-stimulatory domains, usually from CD28 or CD137, can be added to the CAR construct. This leads to increased persistence and superior functionality of these CAR NK cells. CARs from the first generation have no stimulatory domain, whereas CARs from the second generation and third generation have co-stimulatory domain or two co-stimulatory domains respectively. CARs engineering endows NK cells with antigen specificity. The binding of a CAR to tumor antigen delivers a potent activating signal that triggers NK cell cytotoxicity, which results in elimination of cancer cell. As I explained you, through CARs or through specific T cell receptor modification, there are the targeted cellular therapies. Now, these targeted cellular therapies can also be into the variable factors, like these targeted cellular therapies can target the growth factors also. Like I will give you the example here that different two growth factors if we can combine, and one growth factor should be the targeted and manipulated, and combining the two growth factors we can reduce the tumor cell mass. Combining a human epidermal growth factor receptor type 1 or combining the other growth factor as inhibitor, we can use as the targeted cellular therapy. Like in the case of hepatocellular carcinoma, hedgehog signaling is more important in development of cancer or the liver cancer. Now, if we will target along with cellular therapy hedgehog inhibitor, which can be affected with the cellular therapy, and hedgehog inhibitor infusion and cellular therapy infusion can reduce the HCC mass. Other than growth factors, we can combine the VEGF or the another growth factor and the monoclonal antibodies against particular cytokines. Using combination of targeted agents in the patients, we can certainly reduce or induce efficient cytotoxic lymphocytes in the different cancer. The aim of immunotherapies are becoming prevalent other than liver cancer, like the pancreatic cancer or gallbladder cancers also. Other than the cellular therapies, even the clinical trials are focused on whole tumor cell vaccines. Whole tumor cells can be taken out and used as the vaccines. But it is difficult to prepare sufficient number of tumor cells due to the length of culture time and the potential of contaminating it with bacteria or fungus. If in vitro or in the culture we have the tumor cells, it is difficult to expand those tumor cells because tumor cells will acquire the bacterial and fungal infection more than the normal cells in the culture. However, whole tumor cell vaccine is the one of the methods being used or being tried for the tumor mass reduction. I'm explaining you dendritic cells based vaccine. Like from the cancer patient, you can isolate the monocytes from the peripheral blood mononuclear cells, and then you can give the particular cytokines like GM-CSF cytokine and IL-4 and induce these monocytes into the dendritic cells. Now the question comes here, why I can't isolate the dendritic cells from the whole blood and infuse into the patient? Because dendritic cell frequency goes low in the cancer patients, so it's better to generate in vitro dendritic cells from the monocytes, and now these dendritic cells can be transduced with the tumor lysates, tumor RNA and tumor infusions. When these have the antigen they have seen the antigen of tumor, they become as the tumor or the antigen loading dendritic cells which have the peptides of tumors, or they have seen the whole tumor antigen. Now these antigen loaded dendritic cells can be infused back to the cancer patient for the better antigen specificity and generating the T cell immune response in the tumor mass. However, these dendritic cells based vaccines are not much popular in the clinical trials yet. That how we can make the small peptide vaccines. Like the tumor associated antigens can be broken down into the peptides, and these peptide vaccines can be generated. Like giving you the example, MHC class 1 binding peptides are being used for to develop tumor vaccines. These are simple, safe, stable and economical. These are under the clinical trials phase 1 and 2. And what happened in these, how it can be generated. Like for the mutant KRAS or the MUC1 telomerase peptides can be generated as the vaccine. They induce the cytotoxic lymphocytes. Induction of CTLs need peptides derived from the tumor-associated antigens presented on the surface. Now, once they have seen or activate the CTLs, after seeing the antigen, there is increased immune response, increased IgG antibodies, and patient can survive more, and there is a prolonged survival of the patient with these cellular therapies. Therefore, they are being used as therapeutic vaccine approach. I explained you earlier that antigen pulsed dendritic cells or the adoptive T cell transfer or with the CAR T cells, they can be used for the immune or cellular therapies. So students, let's now summarize what we have learned in this module. T cell associated immune therapies combined with cytokines, vaccines and antibodies has achieved significant anti-tumor activity. Although much work has been carried out, however, the clinical results in solid tumors like liver and the lung remain unsatisfactory, and it is reported in many clinical trials. Owing to the immune escape and the dearth of specific tumor antigens, only proportion of T cells can exert the full anti-tumor function. Decades of improvement have added the development of T cell cellular therapy. The distinct advances of each strategy could be utilized to exploit better T cell cellular therapies in a wide range of cancers, including lung and liver cancer. T cell immunotherapy would act as an effective therapy which would reduce the rate of relapse, improve the quality of life, and prolong survival in the near future. Cellular therapy as an adjuvant to surgical resections. T-cell cellular therapy is one of the best cellular therapy. T-cell associated therapies combined with cytokines, vaccines and antibodies, and has achieved significant anti-tumor activity. T-cell therapies could act as an effective therapy which would reduce the rate of relapse, improve the quality of life and prolong survival in the near future. However, autologous DCs used as adoptive therapy also, and DC vaccine or DCs pulsed with tumor cell lysate are in important clinical uses, and in clinical trials in solid tumors it's being tried out. However, their results are not yet very unsatisfactory. Thank you.